Very good evening to the resource person of the day, Mr. Syed Afsar Ali Bukhari, and my team members, Dr. Irfan Abbas, Mr. Srinivas Rao, Mr. Nagaraj, Ms. Vaishnavi, and all the participants. This is the 12th week of the online lecture series called Know Your IP. And we have been making sincere efforts to rope in experts from different fields to address the participants and to pave way for exchange of the knowledge, information, and ideas on the areas relating to intellectual property rights. This week we have another pleasant news coming up. The DPIIT IPR-Chair of Osmania University has designed an exclusive blog for OU IPR Forum on behalf of DPIIT Chair Osmania University, and the chair blog got inaugurated a few minutes ago by the chief patron of the DPIIT Chair and Honorable Vice Chancellor of Osmania University, Professor D. Ravinder. First we shall have a small video of few minutes, maybe 2 or 3, which reflects the inauguration of the blog, followed by introduction of the resource person, Mr. Bukhari, and introduction of the theme. So now let's have a glimpse of the video which reflects the inauguration of OU IPR Forum's blog spot, which will give you an opportunity to share your views, your opinions, your convictions on different and emerging areas relating to IPR. I request our organizers to play the video. Thank you, sir. Thank you, sir. ఇప్పుడు చెప్పమ్మా ఇదేందో ఒక క్షణం స్టార్ట్ అయిందా మీటింగ్ బ్లాగ్ సార్ ఇది డెడికేటెడ్ వెబ్సైట్ సెపరేట్ క్రియేట్ చేసి ఇది సెపరేట్ చేసి లింక్ ఇస్తున్నారు దీనికి ఉస్మానియాకు ఇచ్చారు సార్ ఓకే This is the list of programs we have already done. Acha, okay. Lectures are in video is పెట్టాం సార్. Okay. So everything will be uploaded here. Yes. ఈ రోజు లెక్చర్ కూడా 5 o'clock ఉంది సార్. ఓరి వెబ్సైట్ కూడా అప్లోడ్ చేస్తున్నాం సార్. ఓకే. అప్పుడు మీరు మెసేజ్ ఇచ్చిన ప్రోగ్రామ్స్ కూడా ఉంది సార్. ఓకే. ఇప్పుడు మనకి ఇది ఇట్స్ ఫండెడ్ బై DPIIT sir, Department for Promotion of International Investment. ఇది ఫోరం మీరు లాంచ్ చేసింది సార్. సో ఇప్పుడు ఎవరైనా బ్లాగ్స్ well that was a brief video, and when you visit our IPR website and OU Forum's blogspot, you have so many instructions, and in fact which can be used to post your comments. Now about today's lecture. The topic of the lecture is Biotech Inventions, what is and is not patentable. And we are very happy to have Mr. Syed Afsar Ali Bukhari, patent attorney, and also the registered Indian patent agent, to interact with you on this emerging area of intellectual property rights. Mr. Bukhari is a patent attorney with 1.5 decades of experience of working in biotech industry, and he is a man of action. In fact, he is a rare resource person with equally theoretical as well as practical knowledge in the area of biotechnology patents and inventions. He has defined and implemented defensive and offensive patent strategies for various biotechnology products. Apart from actively advising the senior management of the companies on the intellectual property risks to business activities and also on implementing mitigation measures. Mr. Bukhari has a rich experience of all aspects of patent practice, including drafting, the freedom to operate, patentability analysis, due diligence, contested proceedings, licensing, and patent protection, cutting across various geographies. And academically, Mr. Bukhari holds a master's degree in the science in life sciences discipline and a master's degree in law with specialization in intellectual property rights. And he is also admitted to practice before the Indian Patent Office as patent agent. I will say only one thing about the resource person. He is a person of discipline and principles. I have had an occasion to meet him on a number of occasions in different capacities. And though formally I was in the position of a teacher, I learned a lot of things while interacting with him. Without wasting your time, I present to all of you Mr. Syed Afsar Ali Bukhari, the today's resource person. Over to you, Mr. Bukhari.
So, good evening, everyone, and I hope that all of you are doing great. And thank you, Professor G.B. Reddy, for your kind introduction. I don't think I deserve all the compliments that you have spoken about me. It's my pleasure to be invited to speak in this weekly webinar series organized by DPIIT IPR-Chair of Osmania University. I think they are doing a commendable job because it is a need of the hour in our country, because I feel that the awareness of IP is not so much as we see in the Western countries. So this kind of programs are really helpful in promoting the IP culture in the country. So I'm really looking forward to the next 1 hour or so interaction with all of you. And in fact, I'm reminded of my personal experience some time ago. It's about 8 to 10 years ago regarding the awareness of IP in the country. I was talking to one of my friend, and another friend of his joined us. And after some time, he asked me about what profession I do. And when I told him that I'm a patent agent, he gave me a confused look. And after a few seconds, he said, "Is it something like LIC agent?" It nearly caused me a heartbreak to be compared to an LIC agent. But I think now things have improved in the last few years quite a bit. There is greater awareness. Government is also actively promoting IP, and DPIIT chair, IPR-Chair of Osmania University and other universities are also doing commendable job in creating awareness about various forms of IP. And in fact, I was pleasantly surprised to see some of the episodes of Shark Tank India, where the sharks have inquired with the budding entrepreneurs if they have filed patents or obtained patents on their invention. And those entrepreneurs who said that they indeed have filed or got patents, they got pretty good deal. So at least it's a blessing in disguise for the patent professionals in the country, because a large majority of people now know that there is some species called patent, and if you have that, you are going to make a lot of money. So I think it's something positive is moving in the country, and I hope that in the years to come, so we will have more better awareness. So coming to today's webinar, I'm going to give you a very high-level overview of patenting in the biotechnology-based inventions. This topic is very vast, and I'm only going to give you a very high-level overview of this field is. And I know that quite a few of you who have joined in this webinar may not necessarily be from life sciences or biotechnology background. Unfortunately, I will not be able to avoid using certain technical terms, but I will try to make it as simple as I can. We will look at the large part of my presentation where we are going to extensively be looking at the various types of biotechnology-based invention and what can be patented and what cannot be patented, how this field has evolved over a period of time, and what are the issues or challenges we still continue to face. This kind of topics require more broader discussion, and a lot of jurisprudence has developed in US and Europe, and many countries look up to those to modify their laws, and jurisprudence and the laws that have evolved in those countries influences other countries. So we will have a more broader holistic view of this field, covering the practice in India, US, and Europe in order to give you a broader perspective. So this is just a disclaimer. So whatever I'm speaking today and the opinions that I'm expressing are those of mine alone, and they do not necessarily reflect the views or opinions of the persons or organizations I was or is affiliated with. This is purely an academic exercise for information purpose, and it does not constitute any legal advice. So if you are acting based upon what you hear from me, so you are doing at your own risk. Let's look at briefly about what biotechnology is. Biotechnology, as some of you may already be aware, that it encompasses the use of biological process. It could be a fermentation-based process or other advanced technologies like recombinant DNA technology, antibody humanization, so on and so forth, or the organisms to make or modify the product. So you can clone a gene in a bacterium and make it express the protein of your interest, and finally purify that protein and use that protein for therapeutic purpose. The biotechnology is not something new, although it's a recent phenomena. But in some or the other form, we have been using biotechnology since time immemorial. It is said that as far back as 6000 BC, Sumerians and Babylonians used yeast. Yeast is a fungus, by the way, to make wine. So it's not new. Every one of us are aware of how curd is made. The underlying process that is involved there is a fermentation process, which is nothing but biotechnology process. The soft and fluffy idlis which every South Indian home makes are because of keeping the batter for the required amount of time to enable it to ferment, so that you have those soft and fluffy idlis. So our mothers, grandmothers, and their grandmothers have been using this since several centuries. But modern biotechnology is not so simple as making an idli batter or making a yogurt or making wine. So modern biotechnology involves cutting the gene and inserting a foreign gene into that to make a recombinant DNA, and you can express the desired protein, and ultimately you can use it for some therapeutic use. And the little Y-shaped structure that you are seeing on the right side of the slide at the bottom here is an antibody. So monoclonal antibodies are frequently used in cancer therapies. They are one of the largest selling biotech products currently in the market, and they have tremendous use. The other complex that you are seeing that has a DNA and Cas enzyme, which is docked to the DNA with little two scissors, which are visible on the slide. That's one of the latest technology called CRISPR-Cas9. CRISPR-Cas9. It's a gene editing tool. You can make specific changes at a targeted place in the DNA, and you can switch off the genes which are responsible for causing the disease. And you can literally create designer babies using this technology, because designer babies are no longer scientific fiction. You can literally do that. And this technology was developed in 2012. The 2020 Nobel Prize for Chemistry was awarded to this technology. Of course, some people have misused this technology, which has raised a lot of ethical concerns. One Chinese scientist used this technology to manipulate, to genetically manipulate the human embryo, and raise quite a lot of ethical concerns there. Probably in the subsequent slides, when we get there, we will discuss some of the issues, ethical concerns or moral issues related to biotech-based inventions. So this slide compares how the traditional pharmaceutical field differs with the biotech field. Pharmaceutical substances are very simple. Take the example of aspirin. It's a very small molecule, not at all complex, but when it comes to biotech-based product, they are highly complex. They are multiple times bigger than the pharmaceutical molecules. Insulin by far is one of the simple biotech molecule, but it is 30 times bigger than aspirin. And when you go to further molecules like erythropoietin, more than 150 times, and it adds more complexity because some sugar molecules are attached to this kind of proteins and making it much more complex. And further, you have monoclonal antibodies, which are several hundred times larger compared to the small molecules. And then you can go further up the ladder. You have viruses, which are more complex, and a whole bacterium can also be used as a medicine. Whole cell pertussis vaccine is one example of it. So which adds to much more complexity, because it will be several thousand times more complex and bigger over the small molecules. Well, this is to give you another perspective of the complexity that is involved with the biotechnology-based products. A little portion of the monoclonal antibody has been zoomed in the box, and it's compared to the small molecule aspirin, and you can see the complexity that is involved when you are dealing with the biotech-based molecule. And it is that complexity that creates a lot of issues, not only for the scientific team, but also for the patent attorneys in obtaining patent protection for these kind of molecules. So this slide gives you an overview of what are the different stages through which a product will undergo, finally leading to the commercialization of the product. It takes any time between 10 to 15 years once you have the lead candidate. And if you start from basic research, it can be even more, 20 or 30 years or even more it can take. If you take the example of the mRNA vaccines against COVID, which have been commercialized by Moderna and Pfizer, the proof of concept for mRNA-based therapy was established in the early 90s, and we have the product only now, and you can see the complexity that is involved there. Of course, those approvals were accelerated approvals. Even then, it took so much time. Although for a good period of time, mRNA technology was not the focus of the biotech industry. They were focused on something else. But now that is mRNA-based technology is going to be another revolutionary technology in the way biotech products are going to be made. So while you go on developing your molecule and going through the different stages of development, you have a lot of opportunities to file patent protection on uh, on the on the developments that you make. So during the early development and basic research stage, you have the ability to file patent protection on platform technologies like CRISPR-Cas or recombinant DNA technology. It's pretty old now. Antibody humanization also. By platform technology, I mean those technologies that have broader application uh, over the others. So they are not limited to that. You can use it in specific organisms or on a specific cell. So if you have the opportunity and if you have developed those kind of technologies, you can file patents on that. Those are going to give you very broad protection. And during early development, when you have identified active substances, you can file patent protection on those active agents. It may be an antibody, it may be a protein, or it may be anything else that is derived or cloned. So those patents give you pretty good protection. And as you go along, once you have identified your lead candidate, so you will first test it in the animal models, that is called preclinical development. And once you are assured of some kind of uh, uh, safety as well as efficacy, then you move to the clinical development. You conduct the clinical trial in humans, phase 1, phase 2, and phase 3. While you are in preclinical development, you would have optimized, so you would have developed the process to make the product on a commercial scale and optimized it. And you may also have worked on the early formulation. You can file patent protection on those things. And when you are going into the clinical development, you can file patents on the therapeutic dosage amounts that you have identified to be effective. If you have identified indications that your molecule is targeting, you can file patents on those things. Not every country grants patent on those categories of inventions, but you can get it. We will look at some of those things. And sometimes the formulations you have developed in the early stages may not work also. You may need to tweak certain things and optimize it. You can obtain file patent protection on those inventions. And subsequently also, you can be further improving your product even after you have got marketing authorization or approval of your molecule, and you can file patent protection on those things. So it's a lot of interaction that goes between patent attorneys and the research scientists. It's not just one molecule or one patent you file in the biotech space. There are lot of patents, a portfolio of patents that you develop around your molecule. One of the company, they have a product which is an antibody by name Humira, and in the infringement proceedings, they asserted around 60 patents against the companies who were trying to make copy of that molecule. So it's not unlike the pharmaceutical field, where you have very limited number of patents covering your molecule. The biotech molecules can be covered by lot many patents. Okay, so now let's get into the more substantive issues related to the biotechnology. And biotech inventions may comprise different kind of biological matter. This is just an illustrative list. We will look at each of these in some details going down the slides. And by living biological matter, I mean that those biological matter that that is capable of multiplication by itself, whereas non-living biological matter is derived from the living biological matter. So I will not go through that. So we will look at each of these things. So Microorganism. Let us look at can microorganisms be patented and how this field has evolved with respect to the patentability of microorganisms. Louis Pasteur obtained US patent as early as in 1873. And although the patent was also related to some process claims, but claim number 2 was directed to the microorganism yeast, which is a fungus. So patents were granted in the US as early as 1873. And subsequently also patents were granted on microorganisms which were generally in an inert or inactive form. Subsequently, around 1930, US enacted Plant Patent Act. And at that time, the general understanding that was developed then was that the living biological matter, living substances are not to be granted patent protection. And that was the thinking prevalent at that time. And obviously, there was nothing in the US patent law that would not or that would prevent the grant of patent on microorganism, but that was the general notion after the enactment of Plant Patent Act. Until we go to the Diamond versus Chakraborty case in 1980. So Ananda Chakraborty was a microbiologist working for General Electric, and he genetically engineered a Pseudomonas bacterium, inserted 4 different plasmids into the bacterium, and it conferred on the bacterium the ability to degrade the crude oil in clearing the oil spills on seas and oceans. And since the thinking at the time was that microorganisms are not to be granted patent, USPTO rejected the claims of this patent application. Process claims were granted, but the product claims were denied by USPTO, saying that microorganisms are product of nature, so therefore they are not supposed to be patented. Then the matter went to US Supreme Court, said that microorganism that Ananda Chakraborty has created was not a product of nature, rather it was genetically manipulated, and that microorganism does not exist in nature. It is the handiwork of man, not the product of nature. So USPTO the US Supreme Court set aside the USPTO decision and said that such a microorganism can be patented, and there is nothing in the patent law that prevent granting patents to those kind of inventions. And in that decision, US Supreme Court made this remark, which was most commonly used in the mechanical field, that the subject matter of patent includes anything under the sun that is made by man. So this sort of opened the floodgates for patenting of microorganism in US. And it also influenced the other countries to adapt their laws and enable patent protection to microorganisms. Yeah, so in India also we had a Chakraborty type of moment, so there was an invention for which a patent application was filed before the Indian Patent Office. The invention was directed to preparing a live vaccine for protecting poultry against brucellosis infection. And Patent Office rejected that invention, saying that the end product contained living organism. So the decision of the Patent Office was challenged before the Calcutta High Court, and Calcutta High Court said there is no statutory bar in the patent law, even if the end product contained a living organism, and they can be granted patent protection. This was a little before the Patent Amendment Act of 2002 came into being. But since the Amendment Act, it's very clear that you can always obtain patent protection on the microorganisms, but not on plants and animals per se. Now the question that often arises is, can we patent a microorganism that you have isolated from the natural surroundings, natural environment? Well, it depends upon which country you are talking about, because the practice is a bit different. As far as India is concerned, you will not be able to, under the product of nature doctrine, Section 3C of Indian Patent Act would prevent you from getting patent on isolated microorganisms. But when you go to Europe, you can obtain patent protection even if you have just isolated the microorganism. What is perhaps required to be shown, because European Patent Office follows a little bit more relaxed approach, so long as you show some kind of a technical effect for your invention, that your invention is susceptible to some kind of an industrial application, you can obtain patent on those inventions, even if that invention or that biological material has just been isolated from the environment. So you can, of course, obtain it in Europe. In US, it is a little bit difficult these days after the Myriad decision that came in a few years ago. You would be required to distinguish it from what is existing in nature. If you are able to show that the isolated microorganism is markedly different from what is existing in nature, you should be able to get patent in US, but it's increasingly difficult if it is just an isolated one. Yeah, so there are some other types of biotech subject matters that we can patent: expression systems, cell lines, and hybridomas. They are treated similar to how microorganisms are treated. A biologist understands microorganism in a certain way, but for the purpose of patentability, microorganism encompasses a broad category of biological material. Literally, it includes everything that is unicellular, and it also includes which is non-cellular, for example, plasmid. So they are treated for the purpose of patent law as microorganism. So you can always patent an expression system, which is nothing but carrying a heterologous plasmid into a cell, typically a bacterial cell or yeast or some other organism that you can use it to express the protein of your interest. And these are genetically engineered, so therefore there is absolutely no bar on obtaining patents on this kind of inventions. You can also obtain patent protection on cell lines. These cell lines are typically used for growing the viruses. You can engineer these cell lines, genetically engineer these cell lines to express the proteins or antibodies of your interest. There are several such cell lines. Viro is one, MRC5 is one, CHO is another cell line. So they can be obtained protection. But again, there is a slight differing practice. So long as you have genetically engineered those cell lines, or you have adapted those cell lines to make those cell lines enable to provide a platform for the growth of viruses or for the expression of your protein, you can obtain it. But if it is a mere isolated one, so you will run into challenges across different countries. India, absolutely, you may not be able to. As I said, with respect to microorganisms, about the practice in US and Europe, you will have to show that there is some technical effect before European Patent Office, or you have to show that it is markedly different from what exists in nature for US. Hybridomas are something that also can be patented. Hybridoma is formed by fusing an antibody producing B lymphocyte with the immortal myeloma cell, and they are really helpful in making large quantities of monoclonal antibodies which are used in cancer therapies. Hybridoma doesn't exist in nature because it's a fusion product of two different cells. So definitely you will not have an objection under product of nature doctrine, but increasingly we are seeing is challenging the applicants on other criteria, because hybridoma technology is a little bit old and now it is considered a routine practice. So you will get an objection under inventive step or under obviousness, saying that this technology is so obvious that you can very easily create an hybridoma cell by fusing B cells with the myeloma cells. So therefore, because of obvious reasons, it cannot be patent protected. But there are ways in which you can try to get a patent protection. You can tie that hybridoma to the specific antibody and the variations in the antibody that you have engineered. So plants and animals is another interesting topic. In almost all the countries, plants and animals are not patentable as such, but microorganisms are. And if you have obtained plants and animals generally through an essentially biological process, which is nothing but a natural selection or the normal breeding process which farmers typically use to obtain any plant or animal variety, you cannot obtain patent on that. But otherwise, if you have genetically manipulated the plants and animals, you can obtain patents, but there is some differing practice across different countries. So we will look at that. So this was an interesting case in India. So that went to the Supreme Court. So probably some of you may be already aware of it. It's related to BT cotton technology commercialized by Monsanto. The patent was granted to Monsanto, and patent had process claim as well as claims directed to the nucleic acid. And these are the two representative claims. And if you look at Section 3J under the Indian Patent Act, and these claims, they are patentable because they are not directed to the plants or animals. Here, of course, it is plants. It is directed to a method of producing a transgenic plant. So it is not against Section 3J. And the other independent claim is directed to a nucleic acid sequence, and a nucleic acid sequence is genetically engineered. So therefore, there is no issue with patenting that because it doesn't exist in nature. So patent office rightly granted the patent. The patent was granted to Monsanto, and Monsanto licensed this technology to different companies, and Newswidow is one of the licensee. And there was some dispute that happened between Monsanto and Newswidow related to the payment of royalties. I will not get into the dispute in the interest of time. So as a result of that, Monsanto terminated the license agreement that it granted to Newswidow on the BT cotton technology, and sued Newswidow for patent infringement because they no longer had the permission to use the BT cotton technology. And as it happens commonly in the patent infringement cases, the defendant always file a counterclaim for invalidity of the patent, which Newswidow did. And the Delhi High Court single judge passed an interim order because this was involving a complex technology and the interpretation of law. So therefore the Delhi High Court did not look into all the issues because trial has not begun. The experts had to be examined. So it passed an interim order. The result of the interim order was that it essentially restored the license agreement, and consequently the patent was valid. And both the parties were not happy with the decision of the single judge, and then they filed an appeal before the Delhi High Court division bench. And for some strange reason, division bench came to the conclusion that these claims are not patentable under Section 3J. And in the previous slide, we have seen that those claims are not within the scope of Section 3J. So it was a strange decision, and obviously it had to be challenged, and this went to Supreme Court, and Supreme Court set aside the order of the division bench and remanded the case back to the single judge of Delhi High Court. And this happened in January 2019, and patent expired in November 2019, and we still do not have the decision. Actually, Supreme Court should have seized this opportunity and ruled on this, because this involved a complex issue of technology and law, and it would have helped in evolving an IP jurisprudence in the country, but unfortunately, this didn't happen. So if we look at the US position with respect to the plants and animals, so long as you have non-naturally occurring, non-human, multicellular living organism, you can always patent. The classic case is an onco mouse, the patent of which were granted to Harvard University. Onco mouse carried an activated onco gene that made the mouse susceptible to cancer, and this mouse was really helpful in screening the cancer therapeutic agent. So therefore there was a lot of benefits of this kind of invention. So patent was granted on this kind of animals. And in Europe also, as I said, you cannot obtain patent if it is exclusively related to plant or animal varieties or essential biological processes. But if something is done through genetic manipulation, then you can obtain a patent on that. So transgenic plants or animals are usually made through genetic manipulation. So therefore, you can always obtain patent protection of those transgenic plants and animals in Europe. But of course, in India, we may not be able to obtain patent on the transgenic animal or plants because our Section 3J is worded differently compared to the law in US and Europe. Okay, so let's come to the some of the non-living biological matter, and here it is the nucleic acid sequence, DNA or RNA, which are nothing but the polymer of nucleotides, and nucleic acids, DNA or RNA, they are present in almost every cell. Of course, RNA is present in certain organisms. DNA is present in almost every cell of all the eukaryotes. Of course, there are exceptions. Let's not get into that. So the question was, can we obtain patent protection to these nucleic acid sequences? The reasoning that was generally used in the chemical-based arts was applied here. It was long held in the chemical arts that if you have isolated or purified a substance from the natural surroundings or environment, it is sufficiently transformative, and therefore you can obtain patent on those isolated substances. And the same logic was applied for granting patent protection to the DNA or RNA sequences, because isolating the DNA sequence from the cells requires breaking the bonds of DNA and excising or removing a particular portion of the DNA from the entire DNA. And that was considered to be sufficiently transformative, and therefore it was considered eligible for patent protection. And a particular way of claiming those kind of sequences was evolved by inserting or using the word isolated in the claim language to distinguish it from from the genomic DNA as it exists in the body versus the DNA that is excised or separated from the cells. So a lot of patents were granted, and until we reach to the myriad decision by the US Supreme Court that was in 2013. So the question before, a little bit background, myriad identified certain mutations in the breast cancer gene and found that women carrying those mutations are susceptible to breast cancer, and they went on to develop some diagnostic tests, filed patents on that, and they also filed patents on the DNA sequences that they identified of the BRCA gene. So they obtained the patent because there was nothing that was preventing from getting patent on them. So there were challenges to those patents, and the matter went to US Supreme Court. And the issue before the court was that whether the act of isolating DNA that is separating from the specific gene or sequence of nucleotide from the rest of the chromosome, is it patentable? And the US Supreme Court held in the negative. It said that a mere act of isolation does not render the DNA sequence distinguishable from what is found in nature. But however, a cDNA sequence is patentable. A cDNA is a complementary DNA. It is synthesized in the laboratory. It doesn't exist in nature. So therefore, there is no issue as far as cDNA is patentable. But if you have merely isolated the DNA, even by cutting open the DNA and showing that it is sufficiently transformative, you can no longer obtain patent on those isolated DNA sequences post-myriad decision. And overnight, it questioned the validity of several thousand patents that USPTO had granted then. And coming to the practice in India, isolated sequences cannot be patented because they are considered to be a product of nature. You will have objection under Section 3C, but you can always patent the cDNA. But in my experience, Indian Patent Office practice is not consistent. I have seen cases where Indian Patent Office has granted patents on cDNA sequences as well as rejected claims to the cDNA sequences. So, and the Patent Office manual is also silent on this kind of biotech subject matters. So, and as far as Europe is concerned, there is no issue, even if you have isolated the DNA sequence, so long as you have been able to find a technical effect or a use of that sequence, you can patent those kind of sequences. So these are some other biotechnology-based inventions that you can patent, and these are again genetically modified, whether it is gene construct or genetic vector, and there is absolutely no issues with respect to obtaining patent protection for this kind of invention because they don't have existence in nature. Yeah. So another very important patentable subject matter is protein or polypeptide and their sequences. A polypeptide is nothing but a polymer of amino acids. Several amino acids are linked through a peptide bond to form a polypeptide, and one or more polypeptide form a protein. So a polypeptide or protein sequence is generally represented by amino acid code. It could be a 3-letter or 1-letter code. So as far as modified proteins are concerned, you can always obtain a patent on the modified protein because they don't exist in nature. But if you have just isolated the protein or polypeptide, can we obtain the patent? Again, we have to apply the product of nature doctrine and the differing practice in different countries. In India, if you have just isolated the protein or polypeptide, you will not be able to get patent. But in Europe, yes, you can get a patent so long as you can show that that protein you have identified it for the first time in sufficiently pure form, and it has some kind of a technical effect, or it is susceptible to industrial application. You can obtain patent on those isolated protein, and if you can obtain patent on those isolated protein or polypeptide, you can easily obtain patent on the sequence as well. As far as US is concerned, after the Myriad decision, then you have again the challenge. If you have just isolated the protein, then you may have some kind of an issue obtaining patent if you are not able to show how different it is from your what is existing in nature. Recombinant protein is nothing but the protein which is already existing in nature, and you have just made it through recombinant, means you have cloned the gene of the protein and inserted into an organism, let's say a bacterium or yeast, and you can purify that product and use it in the therapy. But just because you have made that protein through recombinant means does not mean that you can obtain patent on that recombinant protein per se, so long as that protein is already known. But if you are able to show differences between recombinant protein and the naturally existing counterpart, you should be able to obtain patent protection. For example, naturally occurring interferon beta is a glycosylated protein. There are some sugar molecules attached to that protein, but if you express that protein in a recombinant way, it doesn't have those glycosylation, and that affects the three-dimensional structure of the protein. So if you have those kind of variations that you can show from the naturally existing counterpart, so you can obtain patent protection for those kind of inventions. Yeah, antibodies are really hot topic since last few years. So they are nothing but some Y-shaped proteins which are produced by our immune cells, the B cells, when they are exposed to the antigens. And monoclonal antibodies have been frequently used in the cancer therapies, and some of the top-selling biotech products are antibodies. Humira had more than 20 billion sales in 2021. If you look at the structure of the antibody on the left side of the slide, a typical antibody consists of 4 different polypeptide chains, 2 heavy chains. The ones which are shown in blue are the heavy chains, and the ones in the pink are the light chains. So there are 4 polypeptide chains, and the arms of the Y, the upper region of the arms of the Y, contains variable region, and it is that region which determines the binding of the antibody to the antigens. So each antibody has a particular specificity of binding to a particular antigens. And these antibodies can be produced in different ways, and there are different formats in which these antibodies can be made. For example, if you have identified an antigen, you can inject that antigen into a mouse, and mouse will treat this antigen as a foreign molecule and generates an immune response. Nothing but it produces antibody against those those antigen, and you can subsequently isolate that antibody. You can isolate the B cells and fuse it with myeloma cells to make a hybridoma, to make large amount of monoclonal antibodies. So if you have just isolated this kind of antibody from the proteins from the animals, then you may have some kind of an issue with respect to the patentability. Again, the practice differs from one country to another. So, but everything is not lost. You can still get some decent protection. For example, if you look at the image that is shown as B, you can create that kind of an antibody which does not exist in nature. What has essentially been done is that it is the variable region of the antibody which is responsible for recognizing the antigen. So what you can do is that you can replace on the human frame backbone of the antibody the variable region of the antibody that is coming from the mouse, and it will have a specificity to those antigens, and those kind of antibodies are called as chimeric antibody, and you can obtain patent protection for those because they don't exist in nature. And not only that, within those variable regions, there will be certain regions which are hyper variable, which show much more variation. And another advanced technology is antibody humanization. By you can do that, and you can also engraft those hyper variable regions into the human antibody, and you obtain humanized antibody. So and those antibodies can be patent protected because they don't exist in nature. So, but if you have isolated it, you will have some issues. Yeah, so because if you have just isolated, you can have an objection under product of nature doctrine. In India, you will fall foul of section 3C, and in US, after Myriad, it will be increasingly difficult for you to obtain patent protection for isolated antibodies. But if you have just the isolated antibody, as I said, you can either humanize it or create a chimeric antibody of it, and you can obtain patent protection for that. That is absolutely perfect. You can also obtain patent protection by claiming the genetic vector that encode the naturally occurring antibody as such, because you are cloning the sequence of the naturally occurring antibody along with the other sequences like promoter and termination signal sequences. So that's an artificially created genetic construct or genetic vector, and you can obtain patent for that as an alternative. Although it may not give you the broad protection that you may get it if you have the claims to the antibody as such, but that will still be quite decent. And if you are able to identify some changes, and if you make some changes in the antibodies to enhance the binding efficacy of the antibody, or make some changes in the constant region of the antibody, which is nothing but the tail region of the antibody, you can obtain patent protection for that. You can also obtain patent protection on the formulations that you make of those antibodies. You can have more than one antibody as a cocktail and administer it to the patient, and if that is inventive, you can obtain patent protection for those kind of things, kind of a multi-map therapy. You can always obtain patent protection on the methods of producing the antibodies. So if you have isolated the antibody, these are the different ways in which you can go about protecting your invention if you face the issues from the patent office under the product of nature doctrine, US or Europe, or in other countries also. You can apply the same logic. But when we go to the European Patent Office, as I said, you can obtain patent protection even on the isolated antibody if the antigen is new, and if you have isolated an antibody against that new antigen and able to show that there is a technical effect of that antibody, you can obtain the patent irrespective of how you have obtained that antibody. It could even be just an isolated antibody. But if the antigen is already known, and if you make an antibody against the known antigen, you may run into some issues, not under product of nature doctrine because your antibody is new, but under inventive step, because making those kind of antibodies by injecting the antigen or making a monoclonal out of it is a routine process. So therefore you can have objection under the inventive step or obviousness. And we can still obtain some patents on those things as long as we can show some kind of surprising technical effects. If you are able to show that your antibody against the known antigen has improved affinity or improved binding efficacy, improved therapeutic activity, or reduced immunogenicity, then you can obtain patent protection for those kind of antibodies. Increasingly, broad functional claims are objected to. For example, if you are trying to claim an antibody that is binding to X, Y, Z antigen, then you may have some issues because patent law requires you to describe your invention in sufficient manner so that a person skilled in the art would be able to practice the invention when the patent protection expires. So if you do not have sufficient examples to cover the breadth of the protection that you are trying to claim through a functional claim like the one I've shown on the slide, then you may run into objections. So you will be required to narrow down your claims. So these are another categories of inventions on which you can obtain patents. So they are generally not available in a lot of other countries. Let's say, for example, if you have identified a second or subsequent medical use of an already known substance, then you cannot obtain patent protection. At least in India and quite a few countries, but you can obtain patent protection in some of these countries like US, Europe, Japan, Korea, and others. And often these patents are really helpful in giving quite broad, quite a decent patent protection, because the patents on the active ingredients you may be filing in the very early stages, and by the time you commercialize your product, those patents may be soon expiring. And in the absence of those patents, these secondary patents which are targeted to specific diseases, indications, can be relied upon to have a protection for your product. So of course it's a limited protection only in few countries. And a typical example is that during the COVID times, hydroxychloroquine was used to treat COVID patients, although it didn't work. Suppose if it had worked very well, if someone had filed a patent, it was not disclosed in the prior art, then you could obtain a patent on that therapeutic indication of using the hydroxychloroquine for treating COVID patients. So those are the typical claims that you can use to protect your inventions. So since you cannot patent this kind of method of treatment and medical use, but you can always claim patent protection for the in vitro methods, the methods as practiced in the laboratory. You can also obtain patent protection on the diagnostic kits as an alternative. Yeah, you can also obtain patent protection for the formulations that you have developed, and formulations are nothing but your the biological material along with other inactive substances or excipients like stabilizers, adjuvants, and buffering agents, and they also provide decent protections. You may have some objections from the patent office saying that it is just an admixture of different agents, but if you are able to show that there is a synergistic effect, you can obtain patent protection for that. And often patent office can raise an objection that all these excipients are known, and therefore it's obvious to use them, but you can argue that biotechnology-based arts are highly unpredictable. A particular excipient that is used to stabilize a protein or a biological material may not necessarily be helpful in stabilizing another protein. So this way you can argue and obtain patent protection. Yeah, so some of the biotech patentable subject matter can also raise moral or ethical concerns, particularly when your invention is directed to the genetic manipulation of humans or cloning of humans, any kind of genetic manipulation of human embryonic stem cells or human fetal cell lines, or even use of these human embryonic stem cells or fetal cell lines can give rise to moral or ethical concerns. As I mentioned, that the CRISPR-Cas9 technology, which is a gene editing tool, was used by one Chinese scientist to genetically manipulate the human embryo. The experiments were done in secrecy, and the documents were forged to obtain approval, and it raised quite a lot of ethical concerns. And in fact, that experiment also did not yield the desired results, because two babies, twins were born. They were born prematurely, and when their genome was sequenced, the changes, the genetic changes that were intended to be made were not complete. So, and we do not know what kind of impact this can create. So any kind of manipulations of human cell lines, embryonic stem cells, or germ lines create issues. But as far as your invention is related to non-human animals, you obtain patent protection. But again, those patents are judged against these two criteria. We have seen the case of oncomouse, where the patent was granted because it was felt that the suffering that will be caused to those kind of animals will compared to the benefits that you will have, the benefits outweigh the sufferings that will be caused to those animals. So any inventions that would fulfill this criteria, it requires a kind of a balancing act. And if you are able to show that there is an better medical benefit as a result of those inventions, then you can obtain patent for those kind of inventions. I think I am done with my slides. So and I would really thank everyone.
Thank you so much, sir, for your detailed explanation, not only with respect to the legal aspects of patentability relating to biotech inventions, but also about the basics of the subject.
Good evening, sir. Good evening, IPR-Chair Professor, the organizing team, and all the participants. That was a detailed explanation and informative presentation, sir. So first of all, I would like to thank you for accepting our invitation and helping our viewers and participants in introducing few concepts related to organic chemistry and biotechnology. So intellectual property is one of the domains in the law which is firmly associated and attached to science and technology. And one need to have at least one of any of the flavors and qualifications for succeeding in this profession. So biotechnology and organic chemistry is considered to be most complex subjects and to register patents in these inventions. So thank you for explaining all the basic concepts and nicely explaining them through the best possible illustrations, considering the group you are addressing to. And I hope this would also helpful for those who are appearing for the upcoming patent agent examination in the month of May 2022. So your lecture was very informative, sir, and has laid a road ahead for more biological prospecting and discoveries, inventions in the field of biotechnology. I think we need to focus more on this diversified science and technology subjects for the benefit of our participants, so that they can relate the subjects naturally in case if they're not from the science and technology background. Thank you once again, sir, for gracing this special evening today. And this is an auspicious day for us because we have inaugurated our IPR blog also. So we will meet again with yet another interesting topic. Until the next episode, on behalf of DPIIT IPR-Chair OU, I sincerely thank everyone. Thanks one and all. Thank you.
Yeah, thank you so much, and congratulations for launching the blog. So I look forward to reading it. And thank you.